I'll just introduce John for the benefit of the people in the I don't think they can hear me adequately. John is a distinguished paleontologist. He got his PhD in 1987, I think, which was 2 years after me, from UC Davis. John is a paleontologist by training. He's worked all over the world in places like the Huon Peninsula, the Caribbean, Panama, the Barrier Reef, Hawaii. His work is really interesting and important because of the depth he brings to it with the added time horizon of being a geologist and a paleontologist as well as a first-class ecologist. So it's a pleasure to introduce John. Dan?
Thanks, Terry. Who am I talking to? Okay, so as Terry says, I come from a long background of a long temporal framework for my studies. I really think one of the most challenging issues in ecology is the realization that ecological processes often operate on much longer temporal and much broader spatial scales than we normally have the resources to actually study. So to sort of address this, historical work uses time series data that tries to highlight the importance of long-time ecological processes as well as environmental degradation on the ecosystems that we study. So in my talk today, I just want to give you a bit of an understanding about some of the things that we're doing, some of the historical approaches that we're using in my lab here at UQ, my marine palaeoecology lab, and try to broaden the scale of our understanding of community ecology in time, and also the human dimensions of ecological change. So everybody knows or is getting the feeling now that degradation in the sea is large and pervasive. There's basically nowhere you can run, nowhere you can hide, where an ecosystem in the marine realm has not been impacted by humans. It's also been occurring over much longer time frames than we usually tend to think about. And this is an example from a paper by Lotzi and Worm, which was a tree paper showing just the sheer massive declines in many of the larger marine organisms around the world. Some of these organisms represent only 10% of their pristine or natural population sizes. So one of the things that we suffer from is a temporal myopia. We're not really used to looking at longer time frames, and it pervades our management approaches in all ecosystems. And the shifting baseline syndrome is one of the key manifestations of this temporal myopia. We tend to set inappropriate management goals that are based on short-term experience of an environment degraded a long, long time ago. So every time a new generation comes about, the first time I went out to see a reef was in 1970 something, and I thought that was a pristine reef. But in actuality, what I was looking at was a very degraded ecosystem. But this sort of phenomenon, the shifting baseline syndrome, is sort of a subset of a much larger problem. And that is a fundamental mismatch between the temporal scales of observation and management on the one hand, which are our tools and our approach, versus environmental and ecological change on the other hand. So there's lots of processes in the sea that go way beyond the temporal scales that we tend to utilize for management and understanding of ecological processes. Now, what does that mean? Especially for managers, we tend to manage without context. We tend to fly by the seat of our pants. Well, this is the way it was 10 years ago. I knew somebody who saw something 20 years ago. But these aren't really incorporated very quantitatively into our understanding and our approach to management. Because we don't have a temporal time scale, we tend to have no ability to detect chronic or cumulative stressors. And so there's a lot of uncertainty about whether the the phenomenon we're looking at is due to natural causes or whether it's due to human agency. And I pointed to an early debate with the carniform starfish, which was actually trying to understand that, and we still are trying to get a handle on that. Paradoxically, in an age where everybody sort of disses equilibrium, sometimes this sort of approach will encourage equilibrium views of ecosystem dynamics, when in fact they could be, we're just looking at a shorter term sequence in a longer term cycle. So we have to be, also difficulty in setting management goals and assessing the effectiveness of management actions. How do we know when what we're doing as managers is actually having a positive effect? What scale do we use when we don't have a context for understanding the way that ecosystems can fluctuate through time? So basically, we've really massively underestimated the sort of environmental, ecological, and anthropogenic change over long time periods, and that's where marine historical ecology, in a broad sense, sort of comes in, and we try to look at environmental changes over time, ecological changes over time, look at their whether they're correlated or even causative. And this applies to ecosystems over years, over decades, over centuries, over millennia, sometimes even over millions and millions of years. We can gain insights that are useful for us. So, shifting baselines is what I'm going to focus on a bit today. The way significant changes to systems are measured against previous baselines, which themselves may be substantially different from the original state of the system. The goalpost, the bar keeps, in this case, lowering. Instead of raising, the bar keeps being lowered through time. Here's a great example from Lauren Penichan, who's actually doing a sabbatical in our lab this year. Lauren went out and looked at historical photographs from the Florida Keys, and this was a typical sort of catch for a deep sea catch for a fish on a typical day in 1957. And this is the typical day in 2007. And here we have lots of managers going out there and trying to manage this fishery. What is that? What is that fishery? What is natural? What are we trying to manage? Here's an example from the Atlantic column. This is a well-known model of the biomass through time over 500 years of the cod. And it shows fluctuations due to climate, but also exploitation due to fisheries, overfishing. You go to some other places, you might find the cod fishery having a plot between 1970 and 2000 looking like this, which is way down here. And really, what are we actually managing in this context? Here is basically ecological noise. And some managers might get really excited about an increase in the fish stocks between 95 and 96, and then get correspondingly depressed the following year when it drops, when in reality the fishery shouldn't be an open fishery at all. It's long since departed. Okay, well, what about the Great Barrier Reef in Australia, which is what I'm here to talk to you today, a national iconic environmental treasure, but not only environment. The GBR generates about $6 billion a year. Just for scale, that's equivalent to the Australian wine industry, if you were wondering. So we're as important, at least as important as that. But we do know something. We do know something about the GBR. We do know something about declines in coral, because I'm not going to talk about fish anymore. I'm going to talk about corals. And this is a quite interesting paper, amassed an enormous amount of data that the Australian Institute of Marine Science has been amassing over the last 25, 27 years or so. And basically what Len Diath and his co-workers found was basically a 50% decline in coral cover over the last 25 to 27 years. But what happened before 1985? What's going on prior to the 1980s? How do we investigate that? That's shown, I guess, a manifestation of that is shown here, a couple of historic photographs. Dave Wachenfeld at the Great Barrier Reef Marine Park Authority first went out and started compiling some of these older photographs in the literature. and decided he'd like to have a nice jolly, and he went to some of these places up and down the coast and tried to position himself and take pictures in the exact same spots as these historic pictures. And here's one taken near Bowen, where you can see a dramatic difference between around 1900 and around 2000. Dave and Tara Clark, one of the postdocs on the project, is pursuing this in a more quantitative way in several other sites. So to get on to what this talk is all about today, I wanted to give you some results of some work we've been doing over the last 3 years, from 2011 and extending into March 2015. That was funded by the National Environmental, Research Program. Yes. And this is the project that we had categorizing the cumulative impacts of global, regional, and local stressors on the present and past biodiversity GBR. It's essentially a three-pronged approach. We're looking at the historical ecology, the palaeoecology. We're trying to characterize paleoenvironments based on geochemical and other sorts of proxies. And also a big part of it is geochronology, trying to place the environmental and ecological patterns in a temporal context. And all of this is trying to look at improved management on the inner Great Barrier Reef. Several of the co-PIs are here, Jianxin Zhao, Jez Roth. We had 9 PIs on this project from UQ, JCU, and UWA, 3 ECR/postdocs, and 9 PhD students. So I can't show you all the work that's come out of the project today, but I'm just going to try to summarize some of the things that I think you might be interested in. The project, I think, is fairly ambitious in terms of its geographic scope and also the kinds of data that we gathered. We gathered information all the way up from Cape Grenville on the Cape York Peninsula in the north, all the way down to Moreton Bay in the south. The numbers in parentheses show the sorts of data that we were gathering. The data that I'm going to show you today comes from the reef matrix cores in red and the depth assemblages in yellow. And wherever we got reef matrix cores and depth assemblages, we also got information on the life assemblages. So, whoops. How did that happen? Okay, so the whole idea is to try to link up what's going on today with what happened in the recent past with what happened in the ancient past. So we do things like normal ecologists do and go out and lay transects and go diving because it's really awfully hot to spend all your time on fossils and other things. So we gather information on life assemblages using consistent techniques. The depth assemblages, we use grab samples. We dig out some of the substrate or the dead corals on the sea floor and stick them into a calico bag and bring them back. Now this is a funny depiction of a boat on the water, but that's what it is. And these are our fully replicated transects. And for one of these sort of 20-meter transects, we also draw cores into the sediment, into the reef matrix. Now, get out of your head that these are coral cores. They're not coral cores. They might go through some corals, but they're not the typical coral cores that you generally think about with parietes and things like that. These actually go straight down through the reef matrix. We pounded them with brute force. We cap the core once it's in there, anywhere between 2 and 5 meters. We core. We get some lift bags on it. By brute force, get the cores out of the water, swim them up to the boat, and work on them. And just so you can see, I hope I can get out of this. I'll show you a little movie.
You.
Mm-hmm. There you go. Gotcha. Okay. Why is this so blank? It's blank. There's the core going down. It's being aligned at the top by divers at the top. Okay, it's straight. Mark under. There are 5 divers in the water. Lots of fiddling, but you cap the core so that you don't lose all the sediment. You take the core out. You stick the cap there, a couple of lift bags. brute force to bring it out. This is when I generally swim away. But, so it's not too bad once you get the thing going, at the bottom, and then swim it to the bottom. If you want more information on that, you can talk to Jiz sitting there, because he's the master of the master course, as well as Tara. Okay. Now, how do I get this into... I generally use a PC.
Bottom.
Here.
Bottom. Bottom. Keep going. There.
I didn't put that into my timing for the time. Okay. So what do we do when we... why is that doing that? It shouldn't be doing that. What do we do when we get the cores? First thing we do is we bring them back to the lab. Actually, they're stored in cold storage, but I'll get to that in a minute. The first analytical thing we do is find a nice X-radiograph sort of medical facility and ask them very nicely whether we can work at night, and put those through a CT scanner so that we can identify the nature of the reef framework, have a look at sort of what's inside the cores without having to cut them open. But then we do cut them open, we cut them longitudinally, and then we archive one half of the core. We don't use one half of the core, so for all these cores that we gathered, we've got one half that is totally archived, never been studied or used. And then with the half of the core that we're going to study, we divide it into 5 centimeter segments going up the core. So this is our core library at UQ. It's out at Long Pocket. It's essentially a big freezer, sort of refrigerator freezer room. It's kept at a constant temperature of 4 degrees C. And that's so that if we want to do any later analyses, that hopefully we have some compromise of it being not too cold for some things and not too warm for others. So anyway, that's our archive. And we also keep the working part of the core in there as well as we are working on it. So here's a typical core from the Keppel Islands that Mara Lepore has one of our PhD students has extracted. This is the top of the core. It's basically in 3 different segments. Top of the core, middle of the core, the bottom. This one's about 160, about 1.6 meters. These are the uranium series age dates that I'll talk about in a few minutes. But, the stratigraphy is just terrific, right? The 2000, 1975, 1974, 1860, 1486. These are really nicely accreting reefs. There's not a whole lot of... We don't have a whole lot of evidence for storm deposits or reworking or bioturbation, these kind of things. We've tried to target habitats that were similar and weren't subject to these kinds of things. Preservation is absolutely gorgeous. Most of our studies we tend to do to the genus level as opposed to the species level. This is more a function of the time that a PhD student has to complete a thesis as opposed to maybe what we would actually like to get to at some point in time. Because the preservation is so good, we could easily have as much trouble getting into species as modern taxonomists have getting modern specimens to species. But what we do with these, because they're so well preserved, is we can also run uranium series dating on them, collect them from the field, sort through them, look for some clean specimens maybe, stick them through this fancy machine that Jan Shin runs, spends his life worrying about, mass spectrometer, and lo and behold, we get some dates. And the thing that Jan Shin, who's sitting right here, has been developing at UQ that nobody else can do, is that he's able to date very, very young corals all the way back to very, very old corals. And the resolution is just phenomenal. Plus or minus 1 year, plus or minus 2 years. I mean, that doesn't happen for things 400 or 500 years ago, but maybe it's plus or minus 4 or 5 years for things that old. So it's just a great technique for constraining the sorts of patterns that we're interested in constraining. I'm going to focus a little bit today on Acropora, the branching corals, I don't think there's anybody in here that doesn't know what a cropper is or how important it is on the GBR, but I just thought I'd put that up there for you. And I'm going to, first of all, now I'm going to get into the results, but first I'm going to tell you what I'm going to tell you, so then at the end I can tell you what I just told you, so that you don't forget. I'm going to take you through a series of vignettes, and those are going to be centered around three main studies, or three main issues, I think. And that is sort of looking at the whole of the inner Great Barrier Reef scale and looking for variation in ecological history. I'm going to show you a study based on depth assemblages and a study based on the reef matrix cores. Then we're going to look at loss of acropora at regional scales on the inshore Great Barrier Reef. We're going to look at the Palm Islands and depth assemblages there. And then we're going to quickly have a look at more local or reef scale variation in ecological history. And that's going to come from some reef matrix cores in the Franklin Islands. Okay, so let's get to the first bit. And this is a paper that formed the part of Jez Roth's PhD that he was able to publish in the Proceedings in 2013. And we're looking at life and death assemblages from Pelorus Island and the Palm Islands at three different sites. We went and sampled both death assemblages and we cored there as well.
I have no idea.
Looks like some button's pressed.
I think the button is sticky.
I'll just use the right button instead of the sticky.
Oh, giving it.
Oh.
About stuck. Yeah.
Okay. So here's Pelorus Island, a north side, a central or a beach side, and the south side. This is what the area looked like when we were there in 2007. Basically killing fields of acropora in the north and the south side, and abundance of pavona in the central or the beach side. Okay. And here's what we find when we compare the life assemblages and death assemblages. The life assemblages are on the left and in white. The death assemblages are in black and on the right. And these are the three different sites here, A, B, and C, going down the page. And you can see that in all three sites, death assemblages are dominated by acropora. Okay? I'm sorry they're not in the same order up and down, but each one of these large black things bars is acropora abundance. If you go to the life assemblages, that's not the case. The coral community composition is very much different. Still some acropora in side A, but with a whole bunch of other taxa important as well. Pavona is dominating in the middle side, and other taxa are abundant in the side C. Even if you look at the acroporas that are alive in each one of those sites and sort of break them down into growth forms or species groups or whatever, you find that the death assemblages are dominated by different kinds of acroporas than the life assemblages are. So not only is there this transition from acropora to something else, but even among the acroporas that have made it, they've made a transition as well. So that begs a couple of questions, one of which is, when did this sort of change from these dominant acropora in the death assemblage to not? When did that occur? And secondly, does it have historical precedence? So for the first question, we wanted to go and date the death assemblages and have a look at whether we could sort of constrain the mortality in acropora at these sites. And so I just give you a graph of what the expectations might be before we get into the data, so that we don't sort of kind of cheat and say, oh, well, this looks like that. This looks like this. And I've divided it here. This is sort of a hypothetical age range from 1930 to 2010. And then the relative probability of mortality which just represents the weighted averages of all the dates that you're getting. And so what you might expect for past mortality is a spike in the age dates that's followed by a rapid drop, so that you simply don't see the acroporas any longer through time. The orange denotes a normal population turnover through time that's potentially modulated by preservation. And that is, the older you get, the less number of corals will be around to date. So you might expect a curve like something like this for a normally turning over population. And then in the green, I've just superimposed a recent mortality event, maybe bleaching or a cyclone that we've observed in more recent times, and then a significant drop in date. So these are the kinds of patterns and the kinds of interpretations that we might be able to ascribe to those patterns from these ages in the death assemblages. So this gives the a summary of the results at Pelorus Island. This is just the distribution of age dates from the late 1700s all the way up to the 2000s. Most of the acropora dates come from sites A and C, the northern and the southern one. Most of the Pavona dates come from the middle site. Now, what you find in sites A and B is that the relative probabilities point to ages of about the late 1930s and the mid-1940s for the bulk of the acropora mortality. So you don't really see a whole lot of acropora aging past those dates. In contrast, with the Pavona at Site B, where we have lots of live Pavona living now, we see essentially a population that is showing normal turnover. There's no really huge stops and then a long time frame and then a start again. This is what I would tend to think would be a normal population that's turning over through time. So I think what this shows us is that there is a fundamental turnover from acropora to Pavona that occurred sometime in this time period after the 1940s, 1950s. We have a fundamental shift there. Now, this is that data summarized in just a little bit different way, but it's the same graph that you just saw. And I wanted to contrast it with what's happening up at Cape Grenville, way, way north. Some people think of this part of the Great Barrier Reef as really pristine, and we should be looking at these areas for trying to understand what has gone wrong with maybe some of the other parts of the GBR, especially in the inner reefs. So we've been able to get up there a couple of times. We don't have the dates from our most recent expedition up there, which was only a couple of months ago. But we do have some preliminary dates that tend to show that the pattern for the acroporas in the Cape Grenville area is more similar to the pattern that we see for Pavona at Pelorus Island than it is to anything we saw with acropora at Pelorus Island. In other words, the age dates tend to be pretty consistent through time that stem from the 1980s or so. Something that you would expect more from a sort of a normal turnover of acroporas through time. There is some hint of some spikes associated with, there's a couple of cyclones up there in 2005 and 2006 that had some effects on these reefs. And we don't see any really very much older acroporas and a preponderance of ages in the past. We see something that sort of more reminds us of it as a more stable population that's turning over. Okay, so next I want to go to take the same study, the Pelorus Island study, look at the cores, and then compare it with some reef matrix cores that we've got in the Keppel Islands. And again, I want to, before I jump into the patterns and what they mean, again, we want to have a look at what are the potential outcomes and how you might interpret them. So these are cores going down through the sediment, and the sort of the pattern refers to the kind of community. This might be an acropora-dominated community. We might see acropora domination from the very ancient past, 1 or 2 or 3,000 years ago, all the way up to the present. Hey, that's what we're expecting from the cores in Cape Grenville. And that would mean no real sort of imprint of European settlement. On the other hand, we might get sort of stability through time, and then right at the end of the day, sort of the end of the day, in the 80s or 60s or late 1800s, we might expect to see some sort of turnover in the coral communities, from maybe an acropora-dominated community to something else, for example, Pavona, like we saw in Pelorus. Or we might see a pattern of multiple stable states, where there's different coral communities that are characteristic of different time frames, maybe associated with natural fluctuations in climate, and then either capped by this European colonization signal or not. And then the last thing we might see is lots of different states occurring in lots of different order, which means we probably didn't sample well enough, that we'd have to increase our sample size. Luckily, we've spent the money, so we don't have to give the money back. So here's an example in site B of the Pelorus Island. This is basically, the sort of thing that when you see it, you sort of fall off your chair, because what happens is that the cores are dominated by acropora from a long, long time ago. This is the year 266. That's 266 years in the Common Era. So these are calendar dates. And what you see is this transition from domination by acropora in the red to Pavona in the blue. We find that the transition, when you date the very last coral, we're unable to do it here, but when you date the very last coral, the dates coincide almost perfectly with those that we found in the death assemblages for the age of that transition. So here we have really nice evidence in the core that there was, again, this fundamental shift from acropora to Pavona. So, we got excited about this, and you thought, we can really identify what the drivers of this mortality are. We can find a smoking gun. We can raise a whole lot of hell, and try to get people to stop doing what's killing the corals. So we looked at all sorts of things. These are our age dates, the relative probability plots again, frequency and intensity of cyclones, SST anomalies, a PDO index, measures of sediment flux, and the discharge from the Burdekin River. So you can sort of eyeball this stuff, I guess. It's very, very difficult to analyze quantitatively, but you can sort of get a feel that maybe mortality is going up with higher SST anomalies, or maybe there's some higher frequency and intensity of cyclones. These might be associated with a PDO. So mortality, it's always occurring, right? There's always things that are killing corals. But I think a more interesting thing is not really what killed them, but what's preventing them from recovering. And I think this will be a recurrent theme throughout the rest of my talk, is what happened from the 1940s or so, prior to the 1940s, that prevented these things from recovering after the 1940s. Some of the things are a 5 to 10-fold increase in sediment flux from the Burdekin River from about 1870 onwards. So you had a really high increase in the sediment flux to the from the Burdekin. Agricultural fertilizers became widespread from the 1930s, especially when those fertilizers were invented. Suspended sediments, herbicides, nutrients increased. These have been modeled to have increased from 2.1 to 19.5-fold since European colonization. So these are all the kind of water quality issues that may have played a part in the lack of recovery of these corals once they got to a state where they faced potentially multiple mortality events from multiple disturbances, but then the lack of recovery was probably due to these other issues of water quality. Come on. So the question becomes, how widespread is this model? So we've got a case where we've gone from these acroporas to something else, dead fields of acropora. The timing is such that we know that climate change couldn't have been a huge factor in the lack of recovery of these assemblages. How widespread is this? So that provided the motivation really for examining all those sites from Cape Grenville all the way down to Moreton Bay. Was this just a one-off thing, or how does this compare? And of course, there isn't a simple answer to that question. So let's look now in the Keppel Islands to try to see what coring in the Keppel Islands might tell us. And this is from another of the PhD students that was funded on the grant, Mauro Lepore, who's gone through various islands in the Keppel group, and for each of these has gone and done a number of cores at a number of different sites. He's also collected death assemblages, but we'll just have a look at the cores. Now, Mauro's a very careful worker, and we expect a lot more detail from him in the coming months. But the first thing he did was to segregate the branching, the plate, and the massive corals, and provide some graphs that show some preliminary data about the distribution of those growth forms through these cores. And what I've done is I've added these very thick arrows to show all these dates here are uranium series age dates that we have obtained, or that Mauro obtained. And so what I've done is with the arrows is to highlight the cores where we've got dates that span from after European colonization to before European colonization. And if you look at all four of these cores where those big arrows occur, you'll see that pretty much we've been dominated by a very similar assemblage for at least the last 800 or 900 years. So that, and most of these branching corals, Mauro tells me, are comprised mostly of branching acropora. So we don't have any sort of evidence for this a fundamental shift that's associated with the younger aged colonies or the younger aged portions of the core. Now, as an aside, if you look at some of these other cores, you'll see some really interesting evidence for some multiple stable states going on. And we'll be looking very carefully at the ages of these transitions as they occur, the kinds of corals that are involved in those transitions, and the potential environmental drivers that might be key in driving those transitions. Okay, so to sum up this part of the talk, the whole of inter-GBR scale variation, a fundamental shift in community structure, unprecedented in the last 500 to 1,000 years. It characterizes Pelorus Island, occurred around the middle of the 20th century, long before the effects of climate change began to be seen on coral reefs. The increase in sediment flux and nutrient loading to the region following European settlement was probably the ultimate cause for the lack of recovery, not necessarily for the mortality. And this followed a mortality owing to who knows what, but it probably a series of various acute disturbances that the corals were then unable to recover from, given the water quality changes that they were facing. So shifts in coral community structures, such as those occurred at Pelorus Island, don't necessarily typify the full latitudinal range of inshore GBR reefs, and I think that's preliminary data coming out of Mauro's work in the Keppel Islands, I think, shows that. But stay tuned. Okay, so let's go now to the loss of acropora at regional scales on the inshore GBR, and we're going to look at death assemblages from all of the Palm Islands. This is a study that Tara Clark has been, who's a postdoc on the project. She's been looking at different islands in the Palm Island group, did some benthic surveys, showed that there was lots of variation across that group for the live assemblages, but what happens to the death assemblages? So these are plots that plot for 5 of the most important genera. The live coral cover on the x-axis versus the dead coral cover on the y-axis. And you can see for a pretty big disparity between the live and the death assemblages. Acropora is dominant in the death assemblage in grey, but sort of almost absent in the life assemblages. If you look at parietes in Terminaria, they're very much a part of the live communities, but not so much a part of the dead communities. So there's been this, again, this change that is sort of Palm Island-wide. It's interesting because you have to remember that our principal metric, the ones that people get most excited about, the things that make the news, are that there's the changes to the GBR are measured in terms of coral cover. But the kinds of corals that are dominating many of the inshore reefs today are very different from the ones that characterized pre-European colonization. So we're really kind of limited in many ways in what we're looking at. The decadal-scale monitoring programs, they're not providing a complete picture. Presumably the data's there, and it takes a lot of information a lot of effort to go out there and get that compositional data. But I think we really need to do that. But it's still not going to be a complete picture because it's only over the last 30 years or so. When we start looking at historical ranges of variation, we can start trying to understand, we can have a measure of success framework. And what do I mean by that? I mean, we've got a historical context for seeing whether the activities that we're engaged in from a management perspective are actually having a positive impact on the communities in the context of their historical composition. Now, that's not to say we want to go back to some Garden of Eden, that we want to sort of walk the path and really get back to something. That'll never happen. There's lots of reasons why that won't happen. But it does provide a framework for understanding where we are and potentially where we want to get to. Okay, so keeping on with this theme, again, going back to the expectations, because we're not done with our regional analysis of the Acropora loss in the Palm Islands. Just remind you of the expectations slide. And then when you look at places, when Tara looked at Havana Island, Pelorus, and Pandora, she found that the timing of mortality was asynchronous. So here's our Pelorus Island site, which has the mortality concentrated in the 30s, 40s, and 50s. But in Havana, we're here in the 1980s, and Pandora, we're in the 1990s to 2000s. So the mortality is always going to be, the mortality is asynchronous across these reefs. But what you can do, again, is use the timing of that mortality to help you to constrain the drivers. You can help to identify drivers. And I think not only drivers of mortality, but drivers that inhibit recovery, because they may or may not be the same. And I think it's important to ask that question. So the chronic water quality issues associated with lack of recovery in Pelorus may be quite different from the acute disturbance issues that we need to worry about in some of those other islands, although we're going to have to worry about water quality everywhere, obviously in the Palm Islands. Okay, so just a little summary of that vignette. We've got a shifted baseline of what was natural in the Palm Islands. Decadal-scale monitoring programs, they're incomplete. They only span 3 decades, and most of what we can do is based on coral cover. Changes in community composition since European colonization, I think, can be important for predicting future response to environmental change. I think when we look at those shifts in community composition, they can provide predictions, because we've got the environmental information as well, with the geochemical data, with the historic data, the proxies that we use for climate and other kinds of environmental change. We can use those in models to actually predict what the community composition might be, and we can sort of use the past to ground truth those models. I'm really sorry about this, but it wasn't my fault. Okay, so the last little vignette that I'll go over now is reef scale variation in ecological history. Very quickly, some work done in the Franklin Islands using reef matrix cores. This is done by another one of the PhD students on the project, Hannah Markham. And Hannah's gone to 3 islands off of Cairns, just south of Cairns, the Franklin Group, High Island, Normanby Island, and Russell Island. And again, Hannah's work is in progress, but she's been able to identify a disparity between the patterns that exist at Normanby and Russell Island versus those occurring at High Island. She's been able to identify a couple of different communities, a mixed community shown in red, and a branching parietes-dominated community shown in yellow, orange. And what she finds is, again, sort of like Pelorus Island, a transition from one community to the next at 2 of those islands, but at the other island, essentially carried on just as it had been through millennia. And here is some of the dates from that core. Actually, Hannah's cores are a bit shorter. They're about 2 meters, so they go back to about the early 1700s. And again, she's used uranium thorium dating to try to get at when that transition might have occurred. And again, it's sort of middle of the last century to a little bit later, 60s, 70s, and 80s. But again, that's work that Hannah is still progressing on. She's going to get more into the taxonomy and the environmental drivers of that transition. Okay, so to conclude then, you can see that we've got variability at a lot of different spatial scales, variation over the whole GBR, over various regions, between various regions, and even within the same region and the same island group, we've got variability. So it really adds another layer of complexity to trying to understand the shifting baseline on the inshore Great Barrier Reef. We think that a lot of this data points to land use changes since European colonization that have depleted abundance of acropora, that have also affected the stability or persistence of various other kinds of coral communities on these inshore reefs. We know that many drivers kill corals, but really the lack of recovery is what I think is one of the most important things to study, and it appears to us that it's a water quality issue. At impacted sites, historical community composition differs markedly from that derived from present-day and decadal-scale monitoring surveys, so we have to keep that in mind when we're sort of being towed around the reef and we're looking down or we're collecting information on line transects. Is this really what's always been here, or are things been different, and what can we learn from past assemblages? Identifying timing drivers of change is important, but so is lack of recovery. How many times have I said that? So one size does not fit all in the GBR. There's many sources of mortality. Let's focus on the recovery. Whoops. In some sense, we need to get away from the idea of what's the biggest threat on the reef, because the reef is a big thing. It varies over multiple spatial scales. There's different reefs that are having different histories. So I want to I'd rather have the attention focused more on managing the local reefs, managing for the actual drivers of change at appropriate spatial scales. Lastly, palaeoecological studies can provide a quick essay. We can do this for you. Give us $60,000 and go out on the reef. We can take 50 or 100 corals. Cost about as much as a week's ship time on one of the Ames vessels. But I think the information is really worthwhile and worth it to go out and get it. I just want to give you one last slide about future directions, some of the other things that I'm starting to think about with some of this data. These cores. I want to start looking for evidence of historical macroalgal dominance in the reef cores. So not only sort of looking at coral to coral transitions, but maybe trying to get at this holy grail of where there shifts to macroalgal dominance versus coral dominance. And we've started to try to get ancient DNA, do this with an ancient DNA approach from our sediment. I'm working with Alan Cooper down in University of South Australia, who's got a very nice ancient DNA lab down there. Also, understanding the cumulative effects of stressors not just on coral ecology and composition, but also on calcification. So we can use the CT scans from our corals, and we can also pick out lots of different species of corals and look at their calcification through time to see if there's been cumulative effects or effects of cumulative stressors on the abilities for these corals to calcify through time as well. Understanding fishing intensity over time using historical archives and also ichthyolith abundances in the cores. We've begun to look at fish fish remains in the cores. We had an honors student do that this year, and it looks pretty good. And we're going to be able to get some information on community composition or trophic levels anyway. But it's going to take a lot of work, a lot of effort, because they're not very abundant, and it take a lot of work to get to it. But I think we can do it. And oh gosh, you must be joking. That's at the beginning of this one. Oh, no, that's just the trades. Try to develop some models for predicting winners and losers from reduced water quality over time using a traits-based approach. That's another thing that I think would be quite useful. Lots of people are thinking traits-based approaches using predictive models, but I think if we can incorporate some of these past responses and community composition through time into these models, I think we'll be better off in sort of being able to predict community structure through time. There we go. Thank you very much. So do you have any questions? You want to do questions in the JCU first? Do we have...
Yeah, can you hear us?
I don't think anyone has any question.
Okay.
No question.
Question here in UQ? Pete?
If you're looking at the transition as you go from the Crocker to Pervona in reef record, how sort of precisely can you look at any sort of turnover, sort of gradient of change? I mean, could you sort of look at that? If something happens over a decade, would you be able to pick up a change happening over a decade, or would it be 2 decades, or could you sort of resolve that turnover from one to the other?
Yeah, I mean, I think what we've been trying to do is to date as many corals as we can just before the turnover and just after to try to do that. There was a slide I showed that I didn't. It was when I got to the coring, and there was a core there, and it had a bunch of dates on it. Just for kicks, we had about this much worth of core, and we decided to date every coral in that core going up, and it was amazing. It was like, 1885, 1887, 1893. It was just with the resolution, that we can do. So I think it just, we're bound, I guess, by the 5 centimeter, interval of sampling. So if we were to, we could potentially sort of leave them in the core and then do just that, do every single coral through. But it's sort of a chicken and egg thing. We don't really know the changing composition until we get the corals out to do the taxonomy. But it's looking pretty abrupt in the places that we've been able to see it.
Cindy, can we still ask questions?
Yes.
Yeah. John,
we
Dave
have
Yellowby's here.
We have a question here. David Yellowby.
Why do you use the Burdekin and not the Herbert when you're talking about the Palm Islands, particularly Pelorus?
Well, from what I've seen from the aerial photos and the data and the information that I've seen, that has a larger effect, I guess, during the floods than the Herbert. I mean, the Burdekin is one of the largest rivers in the world when it's running, is it not?
In Australia, yes.
Yeah, in Australia.
I think it's because the paleo regular been established for it and goes back.
All right. Okay.
That's
Yeah, that's where Malcolm's stuff comes from as well.
yeah. I've got a question actually.
One more question.
Sorry, John. Scott
You
Smithers.
look here.
Hey, Scott.
How you going, John? I guess, really enjoyed your talk. I guess the thing that I always think about at Pelorus is why hasn't that reef grown up to sea level like at Orris Point? So if the assumption is that the reef hums away and grows happily forevermore from day dot, like I agree with everything you've said, but I guess playing a devil's advocate here, if the assumption is that stuff hasn't been moved away and you've got perfect stratigraphic order, then the question I always think about is, well, why hasn't that reef actually, in the time that a lot of other reefs in near proximity have got to sea level, why hasn't that one?
Yeah.
It's either had to have been growing slowly or it stopped and started sometime in the past, once or several times. So that's the mystery to me.
Yeah.
Well, any thoughts?
Yeah, that one, I mean, remember these cores are at 5 meters
Yeah.
water depth, so they could still be accreting just fine. I mean, I think in many of our other cores we're finding pretty nice straight lines for reef accretion. So those things are still coming up, right? There's nothing to say that they've stopped accreting or stopped growing.
But there are other reefs around there that have come from.
Yeah. In terms of the depth of the reef, I mean, I've seen, they certainly would have had time to get into that accommodation space and fill that accommodation space.
Yeah.
And I've often wondered that in many reefs where I've gone around the world. Why do you have places where you have a deeper kind of reef front? Why hasn't it grown to sea level? And I don't know the answer to that question.
And another example, I think, which I know you're familiar with, is off the front of Orpheus Island, right at the front of the reef flat, there's a big, massive parietes, which must be 4 metres in diameter or something. So, it's been sitting there 5 metres off the reef edge, but it hasn't there hasn't been a cropper that's overtaken it and swamped it in that time frame. And I think, something, and that, obviously, once again, prior to European settlement, it hasn't been overtaken by a cropper. So, I think what you said about it being patchy is the key. Understanding why things are patchy is the difficulty.
Yeah. I agree.
How much variability do you get within a site between your cores, in the sense of the strata? Are there some areas where there's been a bit more sort of mixing?
Yeah. So if you had better eyes and I left that slide longer, you could sort of have a look at that. I mean, obviously, you don't always just get a perfect sort of progression, but if you look at the Palm Islands core, the Pelorus Island cores, we had I think maybe 4 reversals in 50 or 60 dates or something like that. And by reversals I mean where you get something older on top of something younger, sort of thing. So it's been very, very rare, and I think one of the reasons is because we've been targeting leeward reefs, so the leeward side of the reefs in a little bit deeper water depth. We're not up on the reef flats, or we're not in the reef crest zone, sort of thing. So, and that was intentional to get the sort of the very best stratigraphic order that we could get. So it's worked out like much better than we really ever even thought possible. Not really totally sure why, but.
Do you have any kind of explanation as to why the cropper would stop recovering, but then Pavona would do so well?
Yeah. Well, I mean, the only thing I can hypothesize would be that Pavona is less sensitive to those water quality drivers than a cropper is, at least that local population. So whether that's true or not, or whether we should start sort of doing some experiments to have a look. A similar thing occurs in the Caribbean, as you're well aware, with the Agaricia taking over where Cervicornis, the acropora, has fallen by the wayside. It also has to do with, sort of recruitment, who's there first. who gets there, and then you can have historical effects, I think, where you get some taxa that just get in there and that's it, and they just exclude the other taxa because they weren't fast enough. So any number of reasons, I think, would be worth having a look and following up. Does anybody know about Pavona, whether it has different sensitivities to water quality?
It's less sensitive to bleaching.
Less sensitive to bleaching.
Less than a cropper, definitely.